Hello, in this video we will discuss about the light transduction and also we will discuss about the Wald's visual cycle. And Wald's visual cycle means their relation with the discovery of the scientist's name. So Wald's visual cycle is we will also understand about that. So first of all, we should need to understand the introduction through eye. So here is the eye contain different parts. For example, lens, and here is the lens, and innermost part is the vitreous humor, is the fluid-like structure which maintain the eye and also nourish the eye. And on the other hand, the uppermost layer is the sclera, and inner is the choroid, and innermost layer is the retina. Retina contain more layers, but we will discuss also. And here is the optic nerve, which that give the signal to the brain. And here is the cornea, which that is the inner frontal part, is the watery color or transparent color. And here is the posterior chamber, and the interior chamber, and pupil is used for the dilation and constriction for accommodation of light, and the ciliary body is used for the concave and convex shape of the lens on the basis of the vision. So here is the retina, which that is important for the object vision, watching the object. So here is the light reflection and into the retina through a lens. So the retinal layer can be sensory retinal and pigment epithelial retinal layer. This the sensory retinal means the rods and cone cell are present, which that it detect the light intensity. For example, the cone cell is used for the more light, while the rods feel less. So the wall of eye is the sensory retina, is basically between the sensory retina and wall of the eye, the pigment epithelium. So this is the cone and rod, which that is the outer segment and inner segment. And this is the synaptic terminal, which that is attached with a bipolar cell to the nerves, optic nerve, for the transmission of the signal through a transduction. So that is known as the transduction. Transduction means the light will transmit from outer environment to the eye into the brain. So this is the free floating disk are present, which that is very important for the transduction of light. So this is we will discuss, and we are zooming out this picture to understand that inner side is the for bilayer of phospholipid of plasma membrane like structure, and outer is the cell, which that is also contain bilayer of phospholipid of the plasma membrane. And there are many content, for example protein, and here the opsin are present, which that is made up of um, um, 7 monomer. So 7 monomer to become this. And here the 11 cis retinol, that is the vitamin type, vitamin A, which that is bind to become a rhodopsin. And that is why rhodopsin means it produces red color. If you experience about the cat during the night time. So here is the here you can see this, the transducing contain trimeric protein, alpha, gamma, or beta. And alpha unit contain GDP, inactive formation, to become active after the light will be fall on the rhodopsin after the binding with the 11 cis retinol to GTP conversion. And this is the activation of the alpha subunit will dissociate and bind with the phosphodiesterase enzyme are present here. And after the binding with that is the component alpha, beta, and gamma also, and bind with the alpha with GTP to trigger the cyclic GMP. To here is the GTP is used for the guanosine cyclase enzyme used to produce the C cyclic GMP, which that is used for the sodium gated channel open to influx of the sodium for normal inactive formation. But the activation for the light transduction, here the cyclic GMP will be converted into the GMP, simple GMP will inhibit the sodium influx. So that is why the hyperpolarization will be occur. And here the potassium and calcium will be increased. So in this way, you should need to understand the outer inner side, the positive charge more, because the efflux of the calcium and potassium will more. So that is why the you should remember with the hyperpolarization just simply. So the hyperpolarization will trigger the exocytosis of the glutamate containing vesicle as a neurotransmitter. This neurotransmitter will bind with the bipolar cell to trigger the nerve impulse. So after this nerve impulse, which that is lead to due to the glutamate, will bind with the other bipolar cell to the nerve cell, optic nerve. So this is the exocytosis will be occur. So anyhow, here is the pigmented epithelial cell, which that is crossing the light. So in this way, the light will pass in this way and target to the rhodopsin. Target to the opsin will bind with the rhodopsin to become a transductional cascade, and in this way it will be activation. But let's begin to understand the deactivation of this rhodopsin. So the rhodopsin kinase enzyme, which that is activate the arrestin, inactive arrestin to activate by the phosphorylation, that will bind with the rhodopsin to inhibit and detach the 11-cis retinol, the vitamin A. So in this way, here is the alpha, beta, and gamma transducin protein containing trimeric will be inhibit due to the inactivation, and that ultimately inhibit that this inhibition will inhibit the phosphodiesterase enzyme, and the inhibit the guanosine monophosphate GMP, and after the inhibition the guanosine cyclase enzyme will again activate to the cyclic GMP formation, and in this way it will again trigger the sodium influx to repolarization and the rest state. So in this way this mechanism can be understand with these things. So inhibit the vesicle exocytosis. A vesicle will be not bind with the nerve, so the nerve impulse will not occur during the night time. So let's begin to, here is the light transduction was, and that was the light transduction. Let's begin to understand the Wald's visual cycle. So first of all, we should need to understand the vitamin A types. So this is the vitamin A type contain a end of the hydrocarbon with a ring methyl group attachment, and hydrocarbon hydroxyl group containing retinol, and only here is the again hydroxyl group attach retinal retinaldehyde. So in this way the retinaldehyde are present, and here you can see this is the beta carotene. These two rings containing hydrocarbons rings, two rings is known as the beta carotene, which that is present in the carrot. So that is why beta carotene we can say. So here is the retinoic acid also produced. This is the hydrocarboxylic group attached with this vitamin A. So let's begin to understand the opsin. This is the opsin, which that is not attached with the cisretinal, but 11 cisretinal when bind. So this is the type of vitamin A. After the 11 cisretinal will be bind with the opsin. After the opsin binding, this is the binding with the opsin with 11 cisretinal to become a opsin C 11 cisretinal retinal which that is to become our rhodopsin. So after this rhodopsin formation, the light will be fall, and this is the light will be detect. After the light will be detect to form a image formation in the retina, in the fovea region, which that is very important, which that is contain density more rod and cone cell. So the cone cell is more far during the cone cell will more active during the dim light. So here you can see this is a rhodopsin, which that is a basically is the photon will be fall. For example, the reflection of the things of the light, which that is fall, and nerve impulse will occur, and all trans retinal will be dissociate, and opsin again dissociate, And this all type of retinal will be go into the liver. And here liver, inside of the liver, the alcohol dehydrogenase enzyme use. After the using of the alcohol dehydrogenase enzyme in an NADH used to produce oxidize into the NAD plus to produce the all. Here is, you can see, this is the retinal type, and all transretinal formation again in the liver. And after this, in the liver, isomerase enzyme used to produce 11-cis retinol, again this. So the retinol all mean the hydroxyl group are present, which that is the alcoholic group. So after this, here is the rods and cone cell. When entry 11-cis retinol into the rods and cone cell will become a NADH will be oxidized to donate the proton to become a 11-cis retinal. This 11-cis retinal also convert via a retinal isomerase enzyme. So in this way, all transretinal will convert and to become again the cycle will be occur. Thanks for watching. This was the video about light transduction in the visualization and Wald's visual cycle. So that is the basically, I hope you make sense about this. And next video we will discuss about the anatomy of the eye and other things. Thanks.